All right. Well, hey, here we are back for the second portion of this review of the Chapter 15 end of chapter problems. So, what we'll do is begin by looking at number 5 and by realizing I probably need to be considering waxing my forearms or something if I'm going to be doing this too much. Okay, number 5, what's it say? Using the information from problem 4, scientists do a further test cross using a heterozygote for height and nose morphology. Let's see. So again, they're heterozygous for height, so that's your big T, little t, and nose morphology, so that's your big S, little s. We're doing a test cross, so they're homozygous recessive for those characters. Okay. The offspring, tall, upturned. So tall, upturned. That number is 40. Let's see. The dwarf, upturned. Wow, why do I keep saying that? It's like a Daffy Duck. Dwarf with upturned snout. That number is 9. Okay, like the other parent, dwarf with a downturned snout. 42 of those individuals. And tall with a downturned snout. There are 9 of those individuals. So again, like the parents would be tall and upturned, or having dwarfism with a downturned snout. So our recombination frequency is 18 out of 100 matings. So that is 18%. Yay. So it says calculate the recombination and then use your answer to problem 4 to determine the correct order of the 3 linked genes. So if you know the recombination frequencies of different genes on the same chromosome, then that lets you know sort of their relative position with one another. So we know the gene for height and the head appendages of the antennae, it recombined 12% of the time. Antennae and nose morphology, nose shape, recombined 5% of the time. And here we just looked, height and nose morphology recombined 18% of the time. So if we're imagining, like, here's a portion of some theoretical chromosome. Well, the A and S gene recombined 5% of the time. And then the T and A genes, I should say, recombine 12% of the time. And then the T and S genes recombine 18% of the time. So these numbers don't need to be an exact match because these positions are only relative. But that at least lets you sort of see the order in which these genes appear on the chromosome. That's good. Let's see. Now we've got number 6. Number 6, a wild-type fruit fly, heterozygous for gray body color and red eyes. So this is number 6. Again, we've got the gray body color versus recessive would be the black body color. And then we have red eyes, or in the recessive ends up being purple eyes. Okay. So gray body is mated with a black fruit fly with purple eyes. Okay, so we've got a heterozygous condition. And we do a test cross. Let's see. So they give us the results and they want us to calculate the recombination frequency. Familiar story, right? So they could have gray body with red eyes. They could have black body with purple eyes. Those are the 2 parents, right? Or you could have recombination occur. You could have gray body with purple eyes, or you could have black body with red eyes. So the numbers they give us, 721 have this phenotype, 751 have this phenotype, that's 49 and 45. So these individuals are like the parents. So our recombination frequency ends up being 94 out of 15, 66. So recombination frequency is about 6% of the time. All right, so that's fair enough. But then they ask a follow-up here. It says, using information from problem 3, what fruit flies, genotypes and phenotypes, would you mate to determine the order of body color, wing size, and eye color genes on the chromosome? So we have a recombination frequency for body color and eye color. If we flip back to number 3, we calculated a recombination frequency for body color and wing shape. What we don't have, so we have body and wing, we have body and eye. What we don't have is wing and eye, right? So let's look at the matings we can perform. We had the heterozygous wild type with the homozygous recessive individuals. That gave us the body color and eye color, right? That's the one we just did. So that's body and eye, which is, again, number 6. We had body color and wing shape, right? And we did a test cross with that. So that's body and wing. Again, I'm sort of going through what we already know. So that was number 3. And so what's left is testing the wing shape and eye color. And we do a test cross with that. So homozygous recessive. Okay. So that's the test cross we perform. We would get that recombination frequency, and then once we put all these together, the 6 and the 17, then you could do the relative positions just like we did back on this previous problem here. All right. So that was 6. Next, let's look at number 7. Okay, number 7 says, assume the genes A and B are on the same chromosome and are 50 map units apart. A map unit is equivalent to 1% recombination rate. So when we calculated number 3, a recombination frequency of 17%, we would say that's 17 map units. All right, so we know these genes are on the same chromosome. So there's gene B, gene A, and we know that they are 50 map units. So let's say MU for map unit. They're 50 map units apart. All right. So keeping that in mind, what else do we know? An animal heterozygous at both loci is crossed with one that is homozygous recessive at both loci. So again, we do a test cross. What percentage of offspring will show recombinant phenotypes resulting from the crossing over? Well, if you're 50 map units apart, that means recombination occurs 50% of the time. It says, without knowing these genes are on the same chromosome, how would you interpret the results of this cross? If you get 50% chance odds of these genes being inherited together, then you can also interpret that as those genes being on separate chromosomes. So if you hit a recombination frequency of 50%, you don't know if those genes are far apart on the same chromosome or if they're on different chromosomes. So having that recombination of 50% is sort of a magic number or a special number. Alrighty. Looking at number 8. The genes for flower, one controlling blue versus white petals, and the other controlling round versus oval stamens. Okay, so we know blue, big B is going to be blue, and then the recessive is white. And then we had round versus oval for the other shape. What's it saying? They are linked and they are 10 map units apart. Okay, so that means if they're linked, they're on the same chromosome. So there's the gene for flower color, and here is the gene for stamen shape. They are 10 map units apart. So these two genes are on the same chromosome. They're 10 map units apart, which means recombination happens 10% of the time. He says you cross a homozygous blue oval plant. So homozygous blue, this will be your parental generation. Homozygous blue is big B, big B. Homozygous oval is little r, little r. You mate that with homozygous white round. So homozygous white would be little b, and round would be big R, big R. Well, shoot, if that's the case, their offspring are all going to be heterozygous, big R and little r. So those are going to be their offspring. It says the resulting F1 progeny, the resulting F1 offspring, are crossed with homozygous white oval plants. So homozygous white, homozygous oval. Hey, that's another test cross. And 1,000 plants are obtained. How many of each of the 4 phenotypes would you expect? Okay, so let's jot down the possibilities, right? The offspring could be, they could get a big B and be blue. And they could get a big R and be round. So that's one possibility. They could end up looking like the other parent, right? They could get 2 little B's and 2 little R's and be white and oval. But we know recombination happens, so you can get other combos, right? They could have white flowers with round stamens, or they could have blue flowers with oval stamens. They might be unlike the parents. But what do we know? We know the recombination frequency is 10%. And we know that there are 1,000 total offspring. So if there are 1,000 offspring and recombination happens 10% of the time, that means 90% of the time they should look like the parents, right? So that means the ones that look like their parents, that happens 90% of the time, there should be 900 of those things. And then those that are unlike the parents, that happens 10% of the time, and there should be 100 of them. And if we split it equally amongst those possibilities, that means you should have about 450 for each of the dominant conditions and about 50 for each of the recessive. All right. So again, you write down what you know. The genes are linked and they're 10 map units apart. You have this cross creating these offspring, perform a test cross. And you know there are going to be 4 possible combinations of offspring. If there are 1,000 of them and the recombination frequency is 10% of the time, 90% of the time, 900 out of 1,000 offspring should look like one parent or the other. But 10% of the time, 100 out of 1,000 will look like a unique combination of traits from the parents. All right. Now that's living, figuring these things out. Okay, number 9. Getting close to being done here. Number 9, you design Drosophila crosses to provide recombination data for gene A, which is located on the chromosome shown in figure 1512. Okay, Okay, so what do we know? Gene A has recombination frequencies of 14% with vestigial wing locus and 26% with the brown eye locus. Approximately where is it located? Okay, so we need to look at figure 1512. Okay. All right, so here is figure 1512. Hey, got it. Figure 1512. They mentioned brown eyes, it's 104 map units. And vestigial wings. So vestigial wings and brown eyes. That's sort of our landmark here. And then gene A is going to be somewhere in the middle. So let me sort of try and recreate that just a little bit. We have our chromosome. We know that that brown eye gene. I'll write songs about those flies with brown eyes. Brown eyes. That was 104.5 map units. And then they said vestigial wings. Was 67 map units. And we know that gene A, somewhere in here, it recombines, what did it say, 14% of the time with vestigial wings and 26% of the time with brown eyes. So that's 30, 40. If we look at these numbers, that ends up being 37.5. So it seems pretty good the way we have this set up. So gene A should fall somewhere in between here. Maybe roughly a quarter to a third of the distance between the vestigial wing and gene A. So it'll be somewhere in this ballpark. Sorry, I forgot that I adjusted the camera location. Okay. So again, we know from the diagram sort of the locations of these chromosomes, and we know the recombination frequency for gene A. So it's going to be in the ballpark of this position. Alrighty. And then one last problem. This is number 12. Good number to end on. Draw it. Okay, we're going to draw some things. It says, assume you are mapping genes A, B, C, and D in Drosophila. You know that these genes are linked on the same chromosome. Okay, so again, we can just sort of draw a generic chromosome out here. Let's see. We have some recombination frequencies. A and B, the recombination frequency is 8%. Again, I'm writing down what I know. A to C is 28%, or 28 map units. B to C is 20 map units, so this is helpful. A to D is going to be 25 map units. And B to D is 33. Okay, so let's start writing down some of these that we know. We said A to B was going to be 8%, so that's 8 map units. Let's see. A to C is 28, but B to C is 20. So that means B probably falls somewhere between A and C. So let's put C out here. We said B to C was 20, right? And A to C is 28. So hey, math works out. All right, that's good. Now we need to find the relationship between B and D and A and D. A and D are 25 map units apart, but B and D is even further. So that means D is farther from B than A is. So let's throw D over here. A and D, we said it's 25 map units, and B and D is 33. So again, math seems pretty good. So the order of the genes would be D, A, B, C. So again, with recombination frequencies, you can get the relative order. It's not the exact location on the chromosome, but it gets you the relative order of those genes. All right. Hey, we zoomed through that pretty quick. As you work through stuff, let me know. If you have any questions, just bring them in class, and we'll get them answered. Thank you so much for sticking with it.